Anatomy of the urinary system. The urinary system consists of 6 principal organs: 2 kidneys, 2 ureters, the urinary bladder, and the urethra. The primary function of the kidneys is excretion, but it plays more important roles that are less commonly realized. They filter the blood and excrete the toxic metabolic wastes. They regulate blood volume, blood pressure, and blood osmolarity by regulating water output. They regulate the electrolyte and acid-base balance of bodily fluids. They secrete the hormone erythropoietin, which stimulates the production of red blood cells and thus supports the oxygen-carrying capacity of the blood. They helped to regulate calcium homeostasis and bone metabolism by participating in the synthesis of calcitriol. They clear hormones and drugs from the blood and thereby limit their action. They detoxify free radicals. In conditions of extreme starvation, the kidneys help to support blood glucose levels by synthesizing glucose from amino acids. In view of such diverse roles, it is easy to see why renal failure can lead to the collapse of many other physiological functions as well. A waste is any substance that is useless to the body or present in excess of the body's needs. A metabolic waste is a substance produced by the body. Among the most toxic of our metabolic wastes are small nitrogen-containing compounds called nitrogenous wastes. About 50% of these nitrogenous wastes are urea, which is a byproduct of protein catabolism. Proteins are hydrolyzed to amino acids. Then, the amino group is removed from each amino acid. The resulting NH2 forms ammonia, which is exceedingly toxic in the body. Thankfully, the liver quickly converts this to urea, which is a somewhat less toxic waste. Other nitrogenous wastes in the urine include uric acid and creatine. Uric acid is produced by the catabolism of nucleic acids, and creatine is produced by the catabolism of creatine phosphate. These are potentially harmful compounds that must be eliminated. The level of nitrogenous wastes in the blood is typically expressed as blood urea nitrogen, or BUN. The normal concentration of blood urea is 10 to 20 milligrams per deciliter. An elevated number is called azotemia and may indicate renal insufficiency. Azotemia can progress to uremia, which is a syndrome of diarrhea, vomiting, dyspnea, and cardiac arrhythmia stemming from the toxicity of the nitrogenous wastes. Convulsions, coma, and death can follow within a few days. Excretion is the process of separating wastes from the body fluids and eliminating them from the body. It is carried out by 4 organ systems. First, the respiratory system excretes carbon dioxide, small amounts of other gases, and water. The integumentary system excretes water, inorganic salts, lactate, and urea in the sweat. The digestive system not only eliminates food residue, but also actively excretes water, salts, carbon dioxide, lipids, bile pigments, cholesterol, and other metabolic wastes. The urinary system excretes a broad range of metabolic wastes, toxins, drugs, hormones, salts, hydrogen ions, and water. The anatomy of the kidney. The kidneys lie against the posterior abdominal wall at the level of the vertebrae T12 through L3. The right kidney is slightly lower than the left. This is because the space occupied by the large right lobe of the liver located just above it. Rib 12 crosses the approximate middle of the left kidney. The kidneys are retroperitoneal, along with the ureters, urinary bladder, renal artery, renal vein, and the adrenal glands. Each kidney weighs about 150 grams and measures about 11 centimeters long, 6 centimeters wide, and 3 centimeters thick. The lateral surface is convex, and the medial surface is concave, having a slit called the hilum. The hilum admits the renal nerves, blood vessels, lymphatics, and ureter into and out of the kidney. The kidney is protected by 3 layers of connective tissue. First, there's a fibrous renal fascia, which is immediately deep to the parietal peritoneum. This tissue binds the kidney and associated organs to the abdominal wall. 2. The perirenal fat capsule, which is a layer of adipose tissue that functions to cushion the kidney and hold it in place. And 3. The fibrous capsule, which encloses the kidney like a cellophane wrapper anchored at the hilum. The fibrous capsule also protects from trauma and infection. The kidneys are suspended by collagen fibers that extend from the fibrous capsule through the fat to the renal fascia. The renal fascia is fused with the peritoneum anteriorly and with the fascia of the lumbar muscles posteriorly. In spite of all this, the kidneys drop about 3 centimeters when getting out of bed in the morning. In some circumstances, they become detached and drift even lower, with pathological results, such as the condition nephrotosis or floating kidney. The renal parenchyma is the glandular tissue that forms the urine. This appears as a C-shaped segment in the frontal section. It encircles a medial cavity called the renal sinus and is occupied by blood and lymphatic vessels, nerves, and urine-collecting structures. Adipose tissue fills the remaining space in the sinus and holds these structures in place. The parenchyma is divided into 2 zones, an outer renal cortex about 1 centimeter thick and an inner renal medulla that faces the sinus. The boundary between the cortex and the medulla is called the corticomedullary junction. Extensions of the cortex, called renal columns, project toward the sinus and divide the medulla into 6 to 10 renal pyramids. Each renal pyramid is conical, with a broad base facing the cortex and a blunt point called the renal papilla facing the sinus. One pyramid and the overlying cortex constitute one lobe of the kidney. The papilla of each renal pyramid is nestled in a cup called the minor calyx, which collects urine. 2 or 3 of these minor calyces converge to form a major calyx, and 2 or 3 of these major calyces converge in the sinus, forming the funnel-like renal pelvis. The ureter is a tubular continuation of the renal pelvis that drains the urine down to the urinary bladder. The kidneys account for only 0.4% of the body's weight, but they receive about 1.1 liters of blood every minute, or 21% of the cardiac output. All this blood is needed more for the purposes of waste removal than to meet the metabolic demands of the kidney tissue. This gives us a hint of how important the kidneys are in regulating blood volume and composition. Each kidney is supplied by a renal artery that arises from the aorta. Just before or after entering the hilum, the renal artery divides into a few segmental arteries. Each of these further divides into a few interlobular arteries. An interlobular artery penetrates each of the renal columns and then travels between the pyramids toward the corticomedullary junction. Then they form the arcuate arteries, which make a sharp 90-degree bend and travel along the base of the pyramid. Each arcuate artery gives rise to several cortical radiate arteries, which pass upward into the cortex. As a cortical radiate artery ascends through the cortex, a series of afferent arterioles arise. The afferent arteriole leads to a ball of capillaries called the glomerulus. The glomerulus is enclosed in a sphere called the glomerular capsule, or sometimes called the Bowman's capsule. Blood leaves the glomerulus by the way of the efferent arteriole. The efferent arteriole usually leads to a plexus of peritubular capillaries. These are named for the fact that they form a network around another part of the nephron called the renal tubule. The renal tubule resorbs most of the water and solutes that filter out of the blood at the glomerulus. and returns these to the bloodstream by way of the peritubular capillaries. The peritubular capillaries carry it away to the cortical radiate veins, arcuate veins, interlobular veins, and the renal vein. These veins travel parallel to the arteries, having the same names. There are, however, no segmental veins corresponding to the segment arteries. The renal vein leaves the hilum and drains into the inferior vena cava. The renal medulla receives only 1 to 2% of the total renal blood flow, supplied by a network of vessels called the vasa recta. The vasa recta arise from the efferent arterioles of the nephrons that are closest to the medulla. Here, the efferent arterioles descend immediately into the medulla and give rise to the vasa recta inside of peritubular capillaries. Capillaries of the vasa recta lead into venules that ascend and empty into the arcuate and cortical radiate veins. Capillaries of the vasa recta are wedged into tight spaces between the medullary parts of the renal tubule and carry away water and solutes that are reabsorbed by those sections of the tubule. Each kidney has about 1.2 million nephrons. To understand how just one of these works is to understand nearly everything about how the whole kidney works. Each nephron is composed of two principal parts: a renal corpuscle, which filters the blood plasma, and a long coiled renal tubule, which converts this filtrate into urine. The renal corpuscle consists of the glomerulus and a two-layered glomerular capsule. The parietal or outer layer is composed of simple squamous epithelium. The inner layer consists of elaborate cells called podocytes. Podocytes are wrapped around the capillaries of the glomerulus. The two layers are separated by a filtrate collecting capsular space. In histological sections, the space appears as an empty circular or C-shaped space around the glomerulus. The renal tubule is a duct that leads away from the glomerular capsule and ends at the tip of the medullary pyramid. It is about 3 centimeters long and is divided into 4 regions: the proximal convoluted tubule, the nephron loop, the distal convoluted tubule, and the collecting duct. The first 3 of these are parts of one nephron. The collecting duct receives fluid from many nephrons. Each region has unique physiological properties and roles in the production of urine. The formation of urine involves 4 distinct steps. First, the proximal convoluted tubule arises from the glomerular capsule. It is the longest and most coiled of the 4 regions, and therefore dominates histological sections of the renal cortex. It has a simple cuboidal epithelium with prominent microvilli, also called a brush border. The microvilli give the epithelium a distinctive shaggy look. Second, the nephron loop, sometimes referred to as the loop of Henle, is the long, U-shaped portion of the renal tubule found mostly in the medulla. This begins where the proximal convoluted tubule straightens out and dips downward or into the medulla, forming the descending limb of the loop. At its deep end, the loop turns 180 degrees and forms the ascending limb. The ascending limb returns to the cortex, traveling parallel and close to the descending limb. The loop is divided into thick and thin segments. The thick segments have simple cuboidal epithelium. They form the initial part of the descending limb and part or all of the ascending limb. The cells here are heavily engaged in active transport of salts, so they must be highly metabolically active and are loaded with mitochondria. The thin segment has simple squamous epithelium. This forms most of the descending limb, and in some nephrons, it rounds the bend and continues partway up the ascending limb. These cells have low metabolic activity, but the thin segment of the descending limb is very permeable to water. The distal convoluted tubule begins shortly after the ascending limb re-enters the cortex. It is shorter and less coiled than the proximal convoluted tubule. It has a cuboidal epithelium with smooth surface cells with almost no microvilli. The distal convoluted tubule is considered the end of the nephron. The collecting duct receives fluid from the distal convoluted tubules of several nephrons as it passes back into the medulla. Numerous collecting ducts converge towards the tip of the medullary pyramid and near the papilla, where they form the larger papillary duct. About 30 papillary ducts end in pores at the conical tip of each papilla. Urine drains from these pores into the minor calyx that encloses the papilla. The collecting duct and papillary ducts are lined with simple cuboidal epithelium. The flow of fluid from the point where the glomerular filtrate is first formed to the point where urine leaves the body is the glomerular capsule to the proximal convoluted tubule to the nephron loop to the distal convoluted tubule to the collecting duct to the papillary duct to the minor calyx, to the major calyx, to the renal pelvis, the ureter, the urinary bladder, and then exiting the body through the urethra. Not all nephrons are identical. Those close to the medulla are called juxtamedullary nephrons. These nephrons have very long loops that extend as far as the apex of the renal pyramid. The remaining nephrons further from the medulla are called cortical nephrons. These have relatively short nephron loops, dipping only slightly into the outer medulla before turning back, or even before leaving the cortex. Nephron loops are responsible for maintaining an osmotic gradient in the medulla that helps the body to conserve water. Although 15% of the nephrons are juxtamedullary, they are almost solely responsible for maintaining this gradient. Wrapped around each renal artery is a renal plexus of nerves and ganglia. The plexus follows branches of the renal artery into the parenchyma of the kidney. This issues nerve fibers to the blood vessels and convoluted tubules of the nephrons. The renal plexus carries sympathetic innervation from the abdominal aortic plexus, especially the superior mesenteric and celiac ganglia, as well as pain fibers from the kidney en route to the spinal cord. Stimulation by sympathetic fibers of the renal plexus tends to reduce glomerular blood flow and therefore the rate of urine production. Another role of the sympathetic fibers is to respond to falling blood pressure by stimulating the kidneys to secrete renin. Renin is an enzyme that activates hormonal mechanisms for restoring blood pressure. The kidneys also receive parasympathetic innervation from branches of the vagus nerve, but the function of this innervation is unknown. Urine formation and glomerular filtration. The kidney converts blood plasma to urine in 4 stages: glomerular filtration, tubular reabsorption, tubular secretion, and water conservation. As we trace fluid through the nephron, we refer to it by different names that reflect its changing composition. The fluid in the capsular space is called the glomerular filtrate and is similar to blood plasma, except that it contains almost no protein. The fluid from the proximal convoluted tubule through the distal convoluted tubule is called tubular fluid. Tubular fluid differs from glomerular filtrate because of substances removed and added by tubule cells. The fluid is then called urine once it enters the collecting duct. This is because it undergoes little alteration beyond that point, except for a change in water content. The glomerular filtration is a process in which water and some solutes in the blood plasma pass from capillaries of the glomerulus into the capsular space of the nephron. To do this, fluid passes through 3 barriers that constitute the filtration membrane. The filtration membrane is composed of 1. The fenestrated endothelium of the capillary, 2. The basement membrane, and 3. Filtration slits. Endothelial cells of the glomerular capillaries are honeycombed with large filtration pores that are about 70 to 90 nanometers in diameter. Like fenestrated capillaries elsewhere, these are highly permeable, although their pores are small enough to exclude blood cells from the filtrate. The fenestrated endothelium of the capillary is composed of endothelial cells. The basement membrane consists of a protoglycan gel. The basement membrane excludes molecules that are larger than 8 nanometers. Even some smaller molecules, however, are held back by a negative charge located on the proteoglycans. Blood albumin is slightly smaller than 7 nanometers, but it is negatively charged and thus is repelled by the basement membrane. Although the blood plasma is 7% protein, the glomerular filtrate is only 0.03 protein. It has traces of albumin and smaller polypeptides, including some hormones. A podocyte of the glomerular capsule is shaped somewhat like an octopus, with a bulbous cell body and several thick arms. Each arm has numerous extensions called pedicels that wrap around the capillaries and interdigitate with each other. The foot processes, or pedicels, have negatively charged filtration slits that are about 30 nanometers wide between them, creating an additional obstacle to large anions. Almost any molecule smaller than 3 nanometers can pass freely through the filtration membrane into the capsular space. This includes water, electrolytes, glucose, fatty acids, amino acids, nitrogenous wastes, and vitamins. Such solutes have about the same concentration in the glomerular filtrate as in the blood plasma. Some solutes of low molecular weight are retained in the bloodstream. This is because they are bound to plasma proteins that cannot make it through the membrane. For example, most calcium, iron, and thyroid hormones in the blood are bound to proteins that retard their filtration by the kidneys. The small fraction that is unbound, however, passes freely through the membrane and appears in the urine. Kidney infections and trauma can damage filtration membranes and allow albumin or blood cells to filter through. Kidney disease is sometimes marked by the presence of the protein, especially albumin or blood, in the urine. Conditions called proteinuria, albuminuria, or hematuria, respectively. Distance runners and competitive swimmers also experience temporary proteinuria and hematuria. Strenuous exercise greatly reduces the perfusion of the kidneys, and the glomerulus deteriorates under prolonged hypoxia, thus leaking protein and sometimes blood into the filtrate. Glomerular filtration follows the same principles that govern filtration in other capillaries, but there are significant differences in the magnitude of the forces involved.